In order to develop novel drugs and compounds that target tumors in the central nervous system, the accessibility and passage through the blood-brain barrier is highly important. Although the cytotoxicity of the compounds can easily be measured by incubating the cells with the therapeutic molecules, it doesn't really tell anything about the actual delivery to the tumor site in the brain. To address this issue, we developed the following protocol that describes how to recreate the blood-brain tumor barrier in vitro on a dish. The usage of immortalized cells of murine or human origin makes this assay very flexible. It can also be easily scaled up to screen dozens of compounds with high reproducibility. In addition, this method allows both quantification and visualization of the passage and targeting ability of the compounds before proceeding with preclinical models. Therefore, our method partially replaces the animal models by using the pre-established cell lines.
Inclusion of patient-derived brain tumor cells assays the patient heterogeneity, while the use of immortalized cells to establish the blood-brain barrier ensures the reproducibility of the results. Altogether, this blood-brain barrier model offers a really interesting tool to assay the drug diffusion or the delivery of drug vehicles. Visual demonstration of the method is critical because sealing of the cells at the opposite side of the insert requires a manipulation that is not often described in other protocols using similar setup. For instance, obtaining the adhesion of astrocytes.